And thank you for the invitation to be here. And I really, truly enjoyed the first keynote that we had yesterday by Thomas. Since I come more from the physics and engineering background, a lot of this is new to me. But today I would like to share some of the research we do, because I think that maybe some of it you can see some new use of. So I have two main stories for you, but first I would like to kind of share a few slides on where I come from. And I'm heading a center, just like Tom, from the Danish National Research Foundation. So here you can see our motto. We really believe in bringing science to life. I'll come back to that. And then we really want to do better science and to grow better scientists, and also to create new businesses. And here you can see some of our aspirations, that we really try to be open, inclusive, to be ambitious, to make a difference, and to collaborate. So also very welcome to collaborate if you see any of these things of interest. So we were established 9 years ago now, when we got this center from the Danish National Research Foundation, but at the same time we got an additional major grant from a private foundation. And you know, those two grants, one was on sensors and nanosensors that we had been doing for many years, and the other one was on something completely new, using micro-nano devices for oral drug delivery. And I remember I was called to the office of the head of the Danish National Research Foundation, because he was not very happy with me, because you cannot coordinate two of these centers at the same time, because you have promised 80% of your time in both of them. So I was really afraid that he would then ask me to make a choice. Which one would you like to continue? But then he suggested, why don't you just merge them, and then you're only coordinating one center? And I thought that was a brilliant idea. And what happened was that actually a really forced marriage is now a very, very happy marriage, because there's so much cross-fertilization. And honestly, a lot of the sensors that we had been developing over the years were kind of looking for applications, and there were not really any. So now, collaborating with the medical sciences, the pharmaceutical sciences, we got a lot of new inspiration. Since then, we have attracted additional funding so that we now have a broader scope. I would like to highlight the Novo Nordisk Foundation, where we now have a challenge grant, where the idea is to kind of combine everything we do. So the idea is to kind of swallow your own doctor, and to be able to do that, to take samples, to manipulate, to sense, we need some sort of energy. And instead of swallowing batteries, here we try to harvest the energy inside our gut. So to use, for example, the acidity in the stomach or the peristalsis in the intestine. I will not cover that today, it's just to kind of see that we are a bit crazy. So in terms of bringing science to life, it kind of to us covered that we really like to work in the space of life science challenges, but it also covers that we really like to translate what we do. So you'll see some examples of spin-outs that we have generated. So here are some of the companies that have already spun out, and we have 3 more in the making, and I'll squeeze in 2 short stories in the talk today. This is us when we're all gathered. This is kind of our annual gathering. We collaborate with SSI, State and Serum Institute, and University of Copenhagen, Pharmaceutical Sciences. We are from 25 different countries, and we also cover a range of different scientific disciplines, from computer science to pharmaceutical sciences. And if you're interested, you can follow us on LinkedIn. So kind of where do we come from? We do a lot of sensors, like small mechanical devices. We do like to repurpose technologies like a DVD player. You can use that to build cool microfluidics. We build huge optical systems for doing something called Raman spectroscopy. And then you'll see that we do a lot of micro and nanofabrication in our cleanroom facilities and also outside. So I have 2 stories. One story is on how can you use devices to, for example, deliver antibiotics or microbes. And the other one is on how can you take samples from the gut. And then I'll squeeze in a little story in the end about cell growth. So first, the oral delivery part. So we started 9 years ago saying the vision is that we would like to, make some of the drugs available without injections. So our scope is not to do new formulations, but to see if there are new ways using micro-nanotechnologies to get drugs into the body. And the reason why a lot of drugs cannot survive taking it orally is because they will be degraded in the stomach, the acidity in the stomach, likewise for microbes if you try to take those. And then also, if they make it all the way to your intestine, they often need help to attach to the mucus there and to get through the mucus and through the epithelial layer. So we, maybe a little naively, we thought, okay, we're going to make some small boxes, containers, micrometer sized in polymers. We're going to fill them with whatever cargo we're interested in delivering, and we're going to coat it with a lid that can then respond to pH changes, for example, and then it's going to open when we want it to open, and it's going to face the epithelial, it's going to embed in the mucus layer, and then it's going to kind of deliver in one direction. That's not always what's going to happen, but that was kind of our kind of a little mechanistic thinking. Here you can see them. They're around 300 microns in diameter, so it's kind of the same size and it also the same feel as sugar grains. So since then we have moved on to a lot of different device materials and shapes, because it's not always this simple container that's going to do the job, as you'll see. But you can load them with drugs, for example, or any powder formulation, and you can also coat them with these pH-sensitive lids. So we have all that technology in place. As a little side story, if you want to check what you have inside some of these small coated devices, you can do Raman imaging. So it's a spectroscopy technique. It's label-free. So you can just focus the laser beam, and the light that's coming back will give you information of what chemicals you have in there. And that was a technology built in-house by Oleksiy and Roman and applied by Chiara to actually map through the lid of a container, what do I have inside? Where do I have drug? Where do I have polymers? And the guys, they then moved on and started using polarized light. So they could not only see the drug, but they could also see the crystals inside a tablet, for example. And that then started a company that's now called Light Novo, where you can buy Raman spectrometers, huge ones and also very small ones. And they even have a system that's very good for looking at bacteria and identifying bacteria. So back to the story. So when you have coated, loaded and coated your small devices, you can put them into a gelatin capsule, like the size that you would normally use for maybe fish oil tablets. But it of course depends on whether it's for humans or for small animals, and the size will vary. We started first with small drugs like ketoprofen, furosemide, things like that, and it really works. We could see we had a much larger uptake, and also for an extended period of time if we did it in rats. And we could also open up the rats afterwards and just see that, yes, indeed, the microcontainers, they embed in the mucus, and that's probably why we see an extended uptake. So after those first proof of concept, we said, okay, maybe we can also use it to deliver antibiotics, probiotics, and also use similar thinking for taking samples out and analyze later. So I'm going to show you some of that. So first, antibiotics. We loaded, we coated, and then we tested how these devices can actually be used to eradicate bacterial biofilms. And this is done in collaboration with Søren Molin and his group at DTU Biosustain. And we could load with an antibiotic called ciprofloxacin, and then we experimented with different sorts of coatings, some coatings that we knew would attach to the mucus and embed in the mucus layer in the intestine. And it really works. We can eradicate 3 times more bacteria in such a biofilm. It's not in animals, it's just an in vitro test, compared to a bolus dose of huge amount of antibiotics. Later on, we kind of did the same trick together with Tine Raslicht and her group at DTU Food, where they're also very much interested in probiotics. And we thought, okay, normally when they try to seed probiotics in animals, they start, and the same in humans, you start by taking a huge dose of antibiotics, and then you pave the way for the new bacteria to actually settle in. So as an alternative to that, we thought, okay, maybe we can use some of these small containers loaded with antibiotics and coated with a layer that, for example, will not disintegrate until you are in the colon, if that's where you would like to deliver your microbes. So that was kind of the experiment we designed with Tine and with Anders here, you can see, and some others. And it actually works. So you can actually give these small containers to animals, mice or rats. And then here you can see the green bars here. That's if you just give the antibiotics in the drinking water, and then later you try to seed with E. coli that are resistant to this type of antibiotics. And you can see there is a higher colonization in the different types of the GI tract when you use the drinking water, but you also see a nice colonization, and only in kind of the colon part of the intestine when you use the microcontainers. So this is a possibility to kind of locally dose and more distributed dose the antibiotics. So then we thought, okay, if it's working like this, maybe we should try to load bacteria also into these devices. So some of the challenges here are just the same as we saw before with drugs. And these organisms are even alive, which is horrible for us. And a lot of them don't like oxygen. So, and then they need to survive the stomach. They are sensitive to oxygen. And they also need help to actually settle down in the mucus layer in the intestine. So first, I thought we could just buy some formulations of bacteria, probiotics, but Tine said, no, then you don't know what you have. So we had to start just cultivating our own bacteria, or Tine did that, and then we had to figure out how to dry them, spray or freeze dry them, and then put them into the small containers. So first we just had to see, can we actually do all of this, and the bacteria, they are still alive and willing to colonize again when they come out. And we could compare just the LGG and then the spray dried, and it kind of really survived. We also checked after they had been inside the containers, and they are still fine. Here they are then also coated with a lid, as you can see in the end. So in one experiment, Koshit, she made a coating on top of the LGG-loaded containers. It's chitosan, it's cross-linked in a way so that it will not be degraded until they actually reach the colon. So we put them into capsules again, gelatin capsules, feeded them to to rats, and then the big capsule disintegrates in the stomach. The small containers, they come out, and then they can travel further in to your intestine, and then they settle down there. As you'll see, there are two graphs. This one is after 6 hours, and this is after 24 hours. If you open up and you see where do I have bacteria, you can see that they are only in the later part where we had decided the coating to release. So there is the possibility of using the devices here for a more targeted seeding. As an alternative to the containers, Fatima, she started to do electrospinning. I don't know if you've seen this, but it's kind of a way, a little bit like knitting, but you spray out a thin fiber of polymer, and then you can do it in a crisscross manner, so you get something that's looking like this. It's kind of fast to produce, and you can use a lot of different varieties of polymers. So what Fatima did was that she actually took this LGG that we had figured out how to spray dry, and she mixed it into a polymer that will kind of protect the dried bacteria, and then she started spinning, crisscrossing, and she made these layers where the bacteria are kind of embedded inside. And then you get this sheet that you can cut into small pieces. She also did sandwich structures to protect the bacteria even more. So here you see that LGG is in the middle layer, and then you have a sandwich around with a different polymer that is here to kind of protect the bacterial layer. And then you can see it's a 3-layer structure. In here you have your bacteria, and then you have protections on both sides. So together with Priscilla from DTU Food and Tine Raslicht, we then checked, okay, will these bacteria actually survive when they're inside just one layer of sheet? This is LGG pullulan, so that's the blue one. And the green one is just the powder. And then the red one is this sandwich structure, the red curve. And you can see that the viability increases quite a lot when we use the sandwich, and it's not that much worse after 70 days than the powder. So it's fine. So then we moved into a competitive study in rats, comparing just LGG powder and our 3-layer sandwich structure. And we could actually see then that they seed just as well when it's a sandwich structure compared to the powder itself. In 2 regions of the intestine, we could actually see that we had enhanced seeding compared to just the LGG powder, one of them being in the colon. I would, of course, have liked it to be that we were much better at seeding than just the powder, but LGG, apparently, it just grows everywhere, so it's more as a model system to then try with some more advanced probiotics later on. Just as a curiosity, now Fatima and Lasse, they have figured out that you can actually cut through these three-layered materials, just like it's same principle as a cookie cutter. So you can actually make advanced three-dimensional structures with your layered structures inside, so you have bacteria in either of these small devices. So that could be something for the future when you want to deliver probiotics. Before moving into the sampling story, I'll just show you 3 types of devices that have not been used for probiotics yet, but I think they could be, so just to give you an idea of what might be possible. So option 1 is actually, can you co-deliver things? Can you make small devices that can both, for example, deliver antibiotics and probiotics? So we designed these 2-chamber microcontainers that would then release one compartment first, and then a second compartment a bit later. And you can time when they're going to release, depending on the coatings you have on top. So we did this. Here you can see it looks a little bit like a small cake structure. And the outer ring is going to release before the inner ring here. We didn't work with bacteria so far. We worked with 2 different drugs. And you can see one is released before, and then the green one here is the second drug coming out. We did it in animals as well. So we loaded a lot of these small double containers into gelatin capsules, and then we fed them to rats, and then we took blood samples at certain time points and analyzed it to see what do we have. And the first drug, propanol, comes out here after around 1.5 hours, the Tmax, and for the second drug, the furosemide, it comes out much delayed. So there are possibilities of doing local co-delivery if that could be of interest. Second option is to work with foil devices. So this is a completely new device that we have designed. And it actually started because we wanted to deliver insulin through the gut, so an oral type of insulin. And with the microcontainers, it just didn't work. And we realized that the microcontainers are never getting through the entire mucous layer, and it's never getting very close to the epithelium. So we thought, is there a way to kind of press against the epithelium layer? So Lasse and Mehdi and Carmen, they designed what we call a self-unfolding device. It's an elastomer. So it's kind of a polymer sheet with a lot of small cavities where you can load your formulations. And then you roll up this elastomer and put it into a gelatin capsule. When the gelatin capsule reaches your intestine, it's going to dissolve. And as it dissolves, the polymer layer would like to regain its original shape. So it's going to unfold, and it's going to press against the intestinal wall. And it looks very uncomfortable in this piece of tissue, but that's because you don't have any organs around it. Otherwise, in pigs, we've seen it, it's very gentle. It's not making any damage. And now we can actually deliver insulin because we're very close to the epithelium. We have also tried with another peptide. It's an peptide niacin, where we actually can enhance the availability by 380% compared to just delivering it orally without a device. And we can also, afterwards, we can do some CT imaging of the pig intestine to see where are the foils and how are they doing. The only thing that can be a challenge is if the foil by accident opens up in the stomach, it's going to stay in the stomach as it is for now. So that's something we are looking at because, of course, we don't want it to stay there forever. The third option that you might have is to work with something called micro-robots or micro-swimmers. So these are small devices. It's a complete hot field in itself, and we're just there with a few people's activities. But these are small particles that can swim by themselves. One option is to use magnesium particles that you coat with a drug layer, whatever you would like to deliver, but you keep a little hole opened in the end. So magnesium is going to react with your liquid. It could be in the stomach, for example, and then they're going to generate bubbles. And as the bubbles are generated, it's going to swim in the other direction. It's propelled. And that's what we are using. And then you can work just with the micromotors themselves, or you can put them into some of the larger devices. And this is what you see here, that we have filled them into some of our microcontainers. And then they come out of these microcontainers, or it could also be the foil. So you're already very close to the mucous layer, and then you just need something to swim the rest of the way through your mucous layer. So that's what we're doing here. And interesting also is that normally these micromotors, they have a very random walk when they move, but here they're moving in more of a straight line because of the viscosity of the mucus layer. Okay, so that was my 3 alternatives that might give you some new ideas of what you would like to try. I now move on to the gut sampling. And as you probably know, it can be very difficult to take samples from all the parts of the intestine. It's a very invasive method. And that's also why I would say a lot of researchers are just taking fecal samples and working with that. But it would be really nice if we could maybe just make small devices that can take samples where we would like to take samples and bring it out for analysis afterwards. So for us, this started in the micro world. So first activities for us started when Sarvesh and Fatima joined us. Sarvesh, he came from a background in a university where he'd been working a lot with these micro swimmers. And Fatima has a background in tissue engineering. So what they did was to take some of our microcontainers and repurpose them. So they put a propellant inside so that the micromotors, they can swim by themselves. I mean, the whole container is going to move forward. And as it's moving, it's growing a tail because it has some precursors inside to form a hydrogel. So there's a hydrogel tail coming out in the bottom as it's moving forward. And this hydrogen tail is kind of engulfing everything it meets on its way. So if you let it swim in a cell culture, it's going to embed the cells that it's going to meet on its way. So then when we thought, okay, this is cool, so why not try to give such a device to a rat? So this was the next experiment, to actually feed some of these small devices to a rat, and then just wait and see until they come out and take some fecal samples and analyze what did they actually have inside this hydrogel tail. And there was actually a good correspondence between what we could find just by opening up the rat afterwards and what was in the tail of this microcontainer. So looking into feces, looking for microcontainers is not probably the way forward. So we started working with bigger devices. So now these are capsule devices, the normal size of a tablet. And we want to do the same. We want to take samples, bring them out for further analysis. So this is where GAF come in, and it's also in collaboration with Sandra from Tom's center. I'll show her later on. But the first thing is just the device. So our first device is a passive device. We're also working on more advanced devices now. But it's a device that you ingest, and then it's activated. It takes a sample. We need to make sure that it seals completely, because otherwise it's going to be polluted as it travels further down, and then you need to recover it in the end. So how is this made? It's made, for now, it's made by 3D printing. If you look at it, it has a little rubber band around the outer part, and it has a little dissolvable disc here. So your sample will move in, and it's going to dissolve the disc. And when the disc here dissolves, the whole thing collapses, because this is what's holding it, the two parts apart. Before we feed it to animals, we put it into a gelatin capsule. So when the gelatin capsule disintegrates, the fluid will move in, and the whole thing snaps together, and it's closed. And then you can collect the sample. So here is just a little video. You can see the gelatin capsule dissolves. Liquid will move in very soon. And then it collapses, and it has the sample inside. So what we did here with Sandra and Copenhagen Zoo was then, before moving into real animal tests, simply just see if we have the stomach from these animals, and we put the device inside. Will it actually trigger in the real environment? And the samples that we collect, do they correspond to the ones that you can just collect directly by taking samples from the stomach? And when we look at the lemur and baboon here, and just pH, the pH we can sample with our device corresponds to what you would just measure if you took the sample from the stomach directly. The same holds actually if you do 16S sequencing. There's a very good correspondence again between what we see and what you can see if you just collect directly with a pipette from the stomach in this case. We then moved on to pig experiments, where we took our devices, and the devices were made such that they would, or they should, trigger in the stomach. and then come out. So we would be dosing the capsules, more than one, to the pigs. And then we would collect samples. In one pig, it was after just 2 hours, we would open it up. But otherwise, we would look for fecal samples 24 and 48 hours after they had been dosed. And one result is that if you just look for the device after 2 hours in the stomach of the pig, there's a very good correspondence between what you can actually see when you do the sequencing in our tablet compared to opening up the pig itself. If we allow the capsules to travel all the way through and we collect it in the fecal sample, we can see there is some contamination. And that's simply because the seal for now is not good enough, so that GAF has solved now. So that's for the next run, this will be there. But it does trigger, and it does come out, the whole device, and it does take the samples. One thing that's also worth to notice is that in pigs, the gastric emptying varies a lot. So some devices come out super fast, others you have to wait for more than 48 hours, which makes it a little bit annoying, but that's just how it is with pigs. So what we've also started now is to do sampling in the intestine, and the sealing has been solved now. And to actually understand what's going on, we are collaborating with O from Aarhus University. So they have nice hospital settings for actually working with the pigs and to inject some of the capsules and then go down with an endoscope so we can actually visualize what's happening in the intestine. So here you have Gaf and Morten over at O's place, and you have the pig here and the endoscope. I'll just show you the video from a piece of the intestine. So this is in a live pig, and you can see soon it's going to trigger. Yes, so you see it was collapsing and is taking the sample from the intestine. And I think we're going to move out a little bit. So again, we have tested, can we take samples and what's the triggering time of these devices? There is some variation in the triggering time. You can see we get sufficient sample mass. It's around 100 milligrams when we do it in ileum and also in the same in duodenum. So now we are moving into the intestine and we can take the samples now in the pigs. So work in progress. This is not published. So as I promised, I'll just show you 2 slides about what you can do if you're working culturing cells. So you are interested in growing organoids or spheroids, or you want to grow bacteria in vitro. So this is a part of a grant where we are expected at some point to spin out a company, and the guys working on it are very hooked on to do that. You can see they've already made the logo and the vision for the company. So the idea here is that normally when you culture cells, you need to do it in perfusion. At least this is when they're the happiest. So they grow with a liquid flow, and that requires a lot of microfluidics. So what we do instead is that we just kind of hack a normal DVD disc. So you have a disc, but you have engraved channels. You have an inner reservoir where you're going to load whatever growth medium you have. You're having a seeding chamber here where you can grow whatever you like, and the design is going to change if you're going to do spheroids or bacteria. And then you have an outer rim here that is going to be where all your waste is going to end up. And then you start spinning the discs, and then the spinning of the disc will generate the centrifugal forces, so you're driving the liquid outwards. And you don't need any pumps. You just need a very simple spindle motor that can drive your disc. And then you can put a stack of them in an incubator, and then you can start growing. And there would be buffer for normally for 5 days. You don't really need to check on it. And I think, yes. So just to let you know that how did we get the inspiration for this work? We knew about the discs, but actually the antibiotics story that I told you with Søren Molin, this is what triggered this development. Because when we wanted to do the in vitro studies with Søren and Helle, they had a system like this. So you can see it's still microfluidics, and it's a lot of small plastic chips with fluidics. And this is the amount of growth medium you need, because a lot of it is going to be wasted in all the tubing here. So that's why Laura and Stine, they came back and said, we need to do this smarter. We need to do a system that can grow bacteria without all of this tubing and bubble traps and so on, a lot of waste of reagents. So this is kind of how we got started And in the beginning, it was actually only for growing bacteria, and now it has been extended to other activities. So I'm gonna finish now. These were my 2 stories, mainly. One on delivery of different things. It could be probiotics. And the other one, an update on how far we are now with our sampling in the stomach and in the intestine. I would like to thank all the foundations that have made all of this possible. And thanks to this awesome group of people who are making it so much fun to go to work every day. And thanks to you for paying attention to this and for the invitation, Tom.
Thanks very much, Anja. We have time for a few questions. If anyone would like to ask them, put your hands up and we will send a microphone. Lona, can someone get to Lona?
She can shout, I know.
Oh, then the audience can't hear then. So.
So absolutely amazing, Anja. So your sampling devices that
Yes.
go through the gut,
Yes.
could they contain a reagent that would stabilize RNA?
Yes, we are actually working on that now.
So we can do
RNA
you can
later.
do transcriptomics
Yes.
afterwards?
Yes.
Okay.
It's actually now the Gaf and Eva from DTU Food, they have embedded it in the disk. So the thing that collapses at some point. Yes. Yes, we're doing that.
Thomas?
Thank you.
Thank you. Thank you so much. Fantastic. It's a new world.
Just like you present a new world to me.
And a new generation of journals. I never heard about the journal. That's fantastic. I think I only can encourage you to go on, but you are not yet at the end. And in both aspects, delivering, so you're placing at the right spot, and also sampling. Concerning the delivering, as I alluded to yesterday, it's the microbial biogeography which matters.
Yes.
I mean, the human intestine is 7 meters long, and it has not only macro niches but micro niches, and this is selected. And so basic researchers try to understand what brings bacteria that they stay at a certain location. They're only there. So what you have to aim for is placing the drugs, or the nutrients, anything you want to interfere with on the host or the bacterial side to the correct place. At the moment, this is what you do is fantastic, but it's too imprecise to make the real point.
I like the challenge.
But there are ways, we can talk later on, there are ways how to tackle that. And the second thing is, I'm also amazing, and it kills more simple models, because my argument always is that you cannot do this. What people really do is then they do biosections, and Eran Elinav, he is the master of how to do that and take samples along the human gut, but he has Israeli students he pays them, I think, enormous amount of money that they get sampled along the gut to get bacteria out and to understand what they are doing there. So your sampling method is probably also the future, but I really don't understand how this sampling works, because the microbes sit in the mucosa, and actually they sit within the mucosa, and they are not in the fluid somewhere. But what you are sampling is fluid.
Yes, but we have, I didn't show it today, but we also have one that can sample the mucosa. I can show it maybe later.
Yeah, so I think that's fun. I mean, you are on the way. This is fantastic. Thank you. Congratulations.
Thank you.
Great. Well, thank you. Is there another one? No? Oh, over there, Kevin. Last question.
Amazing work. I also feel like I'm looking into the future. So I'm curious. I heard about mRNA, but I was wondering about perhaps inhibitory RNAs or microRNAs, delivering those, especially into mammalian hosts or something.
Yeah.
Have you looked at all at that?
No,
that would
but
be
we were happy to try.
Yeah, that would be cool.
Yes, thank you.
Super. Well, thank you. Thanks again, Anja.
Thank you.
Very kind of you.